Thank you very much for the invitation. It's always a pleasure to be at the RNA Council. So I have 10 minutes to talk a little bit about the bias analysis with long-read transcriptomics. So I will try to kind of be fast. Of course, you all know that long reads are great for the analysis of gene expression and transcripts. And basically, the idea is that with the long reads, you can resolve the contiguity of the transcript, so you don't have to do the assembly that you normally do with the short reads. If we look actually to how the data looks like, this is an example of the longer gas data set. And here what you have is two different samples and the reads that were obtained for a particular gene with different library preparation methods and sequencing platforms. And there is a number of patterns that you can see here. You can see that, well, there is like a global patterns that are found in the two samples, but there are kind of differences. Alternative splice forms at this part of the gene. We can see clearly these differential exon inclusions that are giving rise to alternative isoforms. We can also see something like looks like RNA degradation here, and then there is all over this pattern. They are what we call weak signals that are like intron-detention events of slight differences at the exon boundaries that are kind of more scattered around the signal. And then the question is, of course, if all this diversity is real, or if we have some artifacts there. And this is what we try to explain in the longer gas project, and you have there the reference to look at the conclusion that there were many, but I would like to highlight a number of things. We did this benchmark with a number of species and sample types and library preparation methods. So the lessons that we took from longer gas are different. First is that the long-read technologies do capture the full-length structure of the transcript, but actually much more, as you have seen in the previous example. We saw that many transcript reconstruction algorithms take different strategies that are leading to different results. And we detected that the identification of the novel transcript with the technology, which is something that has been claimed as one of the advantages, is actually quite challenging to do in an accurate way. But for me, what's really, really interesting is to see that individual samples include a high amount of sample-specific RNAs. And this is one of the results of the paper in which we have a number of transcripts that were validated by gene code curators on 6 different samples or different combinations of experimental methods. And what you can see here, and this is the scanty classification, what you have to remember here is that the blue full splice matches, these are known transcripts, and any other color is a novel transcript. So what we saw here is that those transcripts that were common to the 6 samples were the known fraction of the transcriptome. And then we have really quite a number of different transcripts that were sample specific, and those were highly, highly enriched in novel transcript forms. Okay, so this has implications because we have this huge diversity. So this was a transcript that were actually validated by the curators. And one of the questions that we are trying to follow up now with is, what do we do when we have multi-sample data sets? So how do we deal with this diversity? The question that we are trying to answer, how do we go with multiple samples? How do we create our expression matrices? And I'm very glad for the first speaker of this afternoon, because she posed the problem that we are also trying to look at here and see how we address. And is how we construct these transcriptomes when we have multiple samples. And there are like, as it was mentioned before, like 2 strategies, what we call join and call. So basically you have the reads for different samples, you put all the reads together, usually by condition, and then you call a joint transcriptome with all the samples together and putting all the reads together. And the other approach is what we call, this is join and call, and this is call and join. So basically here you have the different samples, you call a transcriptome, so you reconstruct a transcriptome in each of the samples, and then you put them together with some tool that allows this joining. And this is how you have your transcriptome per conditions. And there are advantages of disadvantages of each of the approaches. Of course, if you put all the risks together, you have a high discovery power for detecting novel genes or novel transcripts, but actually it has poor scalability because when the number of reads is increasing, it's going to be difficult to do this. On the other hand, on the colon join approach, you have less reads, so you have less power to discover transcripts, but better scalability. So what we wanted to understand here is like, are there really differences in the calling of transcripts when you do one way or another? So we did this experiment in which we have data from 5 mice. We had the kidney and the brain, and then we were looking at how each of these approaches was performing. So what you have here is these kind of upset plots of the detected transcripts. And basically what we are looking here is like the 5 different samples. These are brain samples, individually, called individually, and then the call and join and the join and call approach, right? And then the intersection of all these. And we did this with our scanting tree methods, but actually also with all, other reconstruction algorithms. And there are a number of things to highlight here. First is the different numbers of reconstructed transcripts that you find one way or another, depending on the algorithm. So here we are going up to 40,000 transcripts, while in other methods are around 8,000. So the way to discriminate transcripts is very different. We already knew that. But then what is interesting here is what we see that is the same as we saw with the longer gas, is that actually you have a huge number of novel transcripts that are identified only when you have the join and call approach. So these are transcripts that are detected because you have many more reads. But then the next categories are those that are found independently. Of that, so that are together in all the samples and all the strategies. And what is interesting here, and this happens basically for all the approaches, is that those that are detected in this way tend to be enriched on these known transcript models, while the ones that are specific for this high amount of reads tend to be novel transcripts. But then we have interesting things. For example, these patterns here, these are transcripts that are detected in single samples. And when you do the call and join, so that you call first and then join together, but not when you put all the reads at the first place and then you try to make the calling. So they disappear somehow. So we are trying to understand what this happens, and this is an example of this. So for example, here we have these reads in one of the samples, and this is one of the transcript that is called, and then together with the rest, this is the result that we have. When we put all the reads together, there is a different resolution of this part of the transcript in which all the reads are collapsed together, and then this is the final transcript that is called. So this transcript that was initially in one of the samples disappears when we do this joining of the reads at the first stage. And this is another example that we have with isoquant approach, which we don't understand. But basically what we have is that for one of the samples in the join and call strategy, we get these shorter transcripts. And when we put all the data together, we have these other transcript results. And this is probably related to the different abundances of reads that is resulting in different decision by the algorithms, in which is the final reconstruction transcript. So basically, here the conclusion is that replication leads to the enrichment of full splice matches, so the known transcripts. This high sequencing depth increases especially the novel transcripts, and but again, these are kind of very sample specific, so we really have to understand whether we want to do that. And then these join and call approaches are not always incremental, and we may have transcript models that disappear from the source transcriptome, and this really affects upscaling of these experiments. And we don't have yet a solution for this problem, but hopefully approaches like that was explained before could help. Another thing that I wanted to mention in the 2 minutes I have is how length bias is affecting also the results and what is the consequences of that. And this experiment that I mentioned before, we sequence actually with different methods, with the isoseq methods in 2 different rounds, but also with this other mass-seq approach of Kinnex, in which you are concatenating transcripts and in nanopore. And you can see here differences in length distribution of the reads that we also knew from longer GASP, but actually also differences in the time of tissue. So the brain sample were longer than the kidney sample in terms of reads, okay? Okay, so we were trying to understand in longer GASP how this affects the transcript reconstruction, but actually what we have found is that this also affects quantification. And this is something that you don't notice if you do a simple correlation analysis. Actually, everything looks to it's correlating perfectly. But when you look at this other kind of plots, okay, I'm almost done. When you, I don't know if you are familiar with this, here you have the length of the transcript as a function of the mean expression in the whole transcriptome. You see these biases that are different depending on the sequencing technology. And this is both PacBio. And then when you see these biases that are different for samples and actually the same, right? So, and this is very much related to this length distribution that I mentioned before. And you also have biases associated, for example, with the GC content. You have a very clear bias on the higher GC content, the higher expression. So that means that we need to envision methods to normalize this data so that we can combine from different experiments and from different platforms. So we are starting to do this. It's not as easy as we thought at the beginning, but we are implementing now methods in which we can take this biased data in terms of length and then apply normalization algorithms that will make the distributions more similar. And with that, I just summarize saying that, yeah, the length bias is still a problem with low-risk RNA-seq, and that we need to develop methods to tackle that. And with that, I want to thank you for your attention and take some questions.
So maybe I didn't understand, but a lot of the plots you showed were counting species.
What?
A lot of the plots you showed were counting how many new species, how many different types of species, right?
Of transcripts, yes.
Of transcripts. What proportion of the transcripts would you say you're confident in? And what proportion falls into these things that you're not confident in?
Okay. So one of the things that we did in longer GASP was to validate experimentally many of the novel transcripts. And we surprisingly found quite a high rate of validation. And I would say that of the novel nodding catalogs, so those transcripts that have novel splice sites, about 50% of them were correct and validated, and 50% were not.
I guess what I'm asking is, if you didn't count species, but counted the number of reads.
Oh, yes.
But you're confident. For example, what proportion of reads are the ones that are known, as opposed to the proportion of reads that are not?
10%.
10% are known. So 90% fall into this thing.